As Tim mentioned, my name is Joel. I'm the Chief Technology Officer at Solid. I also head the Exploratory R&D division of the company, which is focused on high-risk, high-reward, next-generation gene therapies as well as non-gene therapies. We are a public company. These are our forward-looking statements. We founded Solid just about 6 years ago with a single mission, which was to develop promising therapeutics for Duchenne muscular dystrophy in a technologically agnostic fashion. And so while we're very excited about gene therapy, we are building a diverse pipeline of assets behind our lead gene therapy. Nonetheless, today we'll talk about SGT-001, which is our lead program. It is we believe, a differentiated gene transfer program using AAV9 to deliver a dystrophin surrogate, dystrophin being the protein that all Duchenne patients are lacking. Alongside that asset and the unique aspects of it, we've also spent significant amounts of time building a scalable manufacturing process. So before we even dosed our first mice for this program over 4 years ago, we'd already begun scaling a process that we believed could scale to the needs of a Duchenne systemic program. I highlighted that we are a technologically agnostic organization, but very focused on gene therapy, and this is our current pipeline, led by SGT-001. We are developing an antifibrotic program using monoclonal antibody technology that targets a known mediator of fibrosis that leads to delays of the loss of ambulation in Duchenne patients. As I mentioned before, on the earlier side, I'm leading a diverse set of programs focused on next-generation gene therapies, including next-generation promoters through collaborations which we disclosed as well as others, next-generation vector biology, new transgenes, and also non-viral and non-gene transfer technologies too. To discuss SGT-001 and to go a little more detail about that program, every aspect that went into our vector, the transgene, the promoter, and the capsid, were all chosen based off of high selective comparative analysis in vitro and in vivo to pick the ideal components to build an ideal vector. Specifically, the transgene was chosen based off comparative functional analyses in disease-specific models, both large and small. That transgene is called microdystrophin 5 and was recently published in a paper, again by Jeff Chamberlain in Molecular Therapy. The promoter we use to drive high levels of specific muscle expression in skeletal muscle, cardiac, and diaphragm tissue is called CK8, and again was chosen based off comparative analyses, both in vitro and in vivo, to pick the ideal promoter that both allows us to drive high levels, but also be small enough to allow us to really build a unique microdystrophin. Finally, we chose AAV9 because of all the success and the clinical exposure leading to significant transduction in muscle and our different target tissues. SGT-001 is currently being tested in a clinical trial called Ignite DMD. We highlighted recent data from our first cohort dosed at 5 × 10^13 vector genomes per kilogram. And the trial is unique in design in the sense that it is incorporating not only young ambulant children, but also advanced stage adolescent children at a higher age range that are no longer ambulant. And so we disclosed earlier in the quarter that at our 5e13 dose, we saw low but variable levels of expression, giving us the strong rationale to dose escalate as soon as possible. And in our last earnings release as of last week, we highlighted that we received approval from our DSMB, IRB, as well as in coordination with the FDA to dose escalate to a dose 4 times higher, 2 × 10^14 vector genomes per kilogram. And we're now moving forward as quickly as possible with dosing patients in that dose range. As I mentioned before, Ignite DMD is being bolstered by a significant investment early on in scalable manufacturing technologies. We took a small-scale academic adherent process, and every patient in Ignite DMD is being dosed from a process that is now at a 250-liter suspension scale, and that process can scale beyond that should we need to. As I just mentioned, as we move forward, we envision that we will go from a 250-liter scale to well beyond that in order to supply what this disease desperately needs. To talk for just a moment on my last slide about our dose rationale and dose selection, as I mentioned before, we chose a starting dose of 5 × 10^13 vector genomes per kilogram, and that was chosen based on balancing the needs of exploring early safety and efficacy in a very stratified disease cohort population, both young patients and older. And in our preclinical data sets, while we saw low levels of expression at that 5 × 10^13 level, we also saw significant variability. This is something we highlighted when we shared our data. Nonetheless, we also described a phenomenon phenomenon that is well known in the gene therapy landscape of a threshold effect, that as you increase in dose, you begin to see more than linear increases in biodistribution. So from a pharmacodynamics perspective, detectable amounts of DNA in tissues. And that correlated with a nonlinear or more than linear increase in protein expression as well. Finally, mirroring all of that transgene data from a DNA perspective and protein perspective, we also saw significant functional benefits and gains at these higher doses as well. And so we're moving to a dose, as I mentioned, that is about 4 times higher than our starting dose in cohort 2, and we look forward to moving very quickly to dosing patients with that current dose level. Thank you.
Thanks, Joel. That's a good update on Solid's progress with the microdystrophin gene therapy program. You know, my first question is obviously, what additional details can you provide in terms of your ability to dose escalate? How quickly can you do it? How many additional patients are you looking to dose this year?
Yeah. Can you hear me? Is this on? Yeah. Great. What we disclosed is that at 2E14 that we had vector on hand to begin dosing immediately, and we actually have sufficient materials to dose quite a few patients at that dose. From a scaling perspective on the gene therapy manufacturing front, we've always envisioned going well beyond 5E13. This was a dose ascending trial from the beginning, and so our estimates and our production capacity was always mirrored by a much higher dose than 5 times 13 in that 2E14 range. And so we're moving very quickly towards dosing and hope to be able to share data as soon as possible.
Is it possible that you may be able to go even higher on the dose, given the fact that I think Pfizer's moved up to 3E to the 14?
Yeah. So our IND-enabling work explored doses as high as 7 times 10 to the 14th. We're very confident that 2 times 10 to the 14th, we should be in a very comparable range to our competitors. And you have to remember that in general, neuromuscular gene therapies have a strong and long history of dose escalation. Across the ranges, we've seen significant efficacy, all predicated by doses that had to be escalated and explored in human trials.
And I think there's one key differentiating item with your gene therapy vector. It's the nNOS binding domain. Could you talk a little bit about that? Yeah. How that might play a role in showing even higher levels of microdystrophin expression?
Sure. As I mentioned before, every aspect of our vector was chosen based off of comparative in vivo analysis and utilizing the latest understandings of dystrophin biology. And so by using a promoter that is small enough in size, yet driving high levels of expression, that allowed us to really tailor suit an ideal transgene, an ideal microdystrophin. And so this microdystrophin incorporates a domain that was only discovered less than 10 years ago, the nitric oxide binding domain. And so in healthy muscle, nitric oxide is localized, or nitric oxide synthase, the protein that produces nitric oxide, is localized to the muscle membrane. And in periods of high energetic demand, nitric oxide is produced. That leads to vasodilation and meeting the oxygenation demands of an exercising muscle. And for the longest time, our collaborators, Jeff Chamberlain and Dong Sheng, had been developing these microdystrophins, but they couldn't find the region within dystrophin that restored this key protein. And so Dong Sheng discovered it, as I mentioned, about less than 10 years ago, and showed in comparative in vivo work that by recruiting that domain, incorporating it into the construct, it restored nitric oxide production, and it led to significant reductions in fatigue in animal models. We've been able to show in our animal data that by incorporating that domain, we see very clear nitric oxide localization as well as nitric oxide production and efficacy.
And I think, what are the next steps? When can we expect to see data at the higher dose? Obviously, I think some of your competitors out there have also moved to higher doses and they're enrolling pivotal studies. Seems like there might be more readouts in the second half of the year. Is that something that's possible for Solid?
That's right. Well, a couple different factors, and we hope to share data as soon as we can. The clinical trial is unique for a number of reasons. I mentioned incorporation of ambulant as well as non-ambulant children, but also alongside every patient dosed is a second patient. It's an open label trial, but there's a second patient enrolled in a delayed intent to treat cohort, and that patient is dosed approximately a year after his counterpart had been dosed in the trial. And what that allows us to do in the context of our trial is really understand on a very granular level differences and deviations from average or from normal in the untreated population. So as I mentioned, we hope to move forward very quickly, and it's important to note that while we've disclosed 3 patients dosed that was incorporated in our efficacy data, that was a total of 6 patients enrolled in the trial, and those delayed intent to treat cohort patients will also be dosed as soon as possible.
I think in your presentation you also highlighted the nonlinear increases that you saw in the preclinical studies.
That's right.
And how that should translate in the clinical studies. Would you just talk a little bit more about that as well?
Yeah. Funny enough, I was just at a conference with a bunch of KOLs in the space, and we talked extensively about threshold effects and liver sinks. You know, 5 years ago when we started developing this program, I remember sitting in Jeff Chamberlain's office and him drawing on a chalkboard an S-curve, something that he saw in his own preclinical work for decades at this point, suggesting that there was a non-linear increase in protein expression that really didn't follow a normal curve, and that 2-fold increases in the dose would lead to dramatic increases in protein expression. You know, we carefully selected our starting dose based off of a large composite analysis of all the preclinical data we had generated across many different species. As I mentioned, that low dose was selected based on wanting to understand very early on in our trial what a 5e13 dose meant, not just in young boys, but also in advanced stage patients where we hope to preserve cardiopulmonary function. So what we saw were low levels of variable expression, and we want to go up from there based on all the modeling suggesting that a nonlinear or a 4-fold increase in expression or a dose should lead to a fairly robust increase in protein expression.
And at this point, the Data Safety Monitoring Board, they're comfortable with the safety profiles that they've seen in the 3 boys. That's
right.
That's mainly the reason why they're allowing dose escalation to occur, right?
That's right.
So, you know, what can you say about the amended protocol, right? So, you know, obviously you had to change the protocol. Steroid treatment is now part of the protocol. You know, do you think that that will provide a good safety margin for SGT-001?
So it's a good question. In our first patient, we highlighted that we experienced a serious adverse event associated with a decrease in platelets and red blood cell count. Our clinical trial PI also identified that complement was activated in that patient. And so based on the composite of the phenotype being presented, which was all biochemical, the patient was doing well throughout the entire experience, he decided to dose the patient with eculizumab. And what we saw when we went after dosing with eculizumab was a fairly quick resolution of all these biochemical features. But when we look back at the data in retrospect, that increase or resolution of those biochemical factors even began before using eculizumab in that patient. And so all patients, even before, all patients enrolled were receiving steroids. The modification of the trial included an intravenous dose of steroids shortly before dosing and for a brief period after. The other amendment to the protocol was to begin to explore children before moving back into adolescence.
Okay, great. I guess for the sake of time, maybe we have one more question. When you look at the manufacturing landscape, and obviously that seems to be a challenge that investors are putting a lot of focus on.
That's right.
How comfortable are you with ramping up manufacturing for your vector? You know, obviously it seems like each company is taking a different approach.
Yep.
You know, how do you see the approach for Solid going? Will it be a hybrid approach, or would you at some point consider having your own in-house manufacturing?
So we're always exploring strategic opportunities around when the right time to build our manufacturing facility would be. You're absolutely right. The gating key in this competitive landscape will be being able to supply drug. We're talking at doses in the 2E14 range, and we're talking about patients that are on average are in the 45 to 50 kilogram range. These are large patients with high doses. And so the process that we've developed at every stage of development has been comparatively analyzed back to the previous scale. In other words, when we switched from adherent to suspension processes, we did extensive in vivo potency analyses to show very clear comparability, and we continue to do that at every step of the process. We also were able to maintain yields at every step of the process. And so as we went up in scale, we saw very clear maintenance of the yields being produced at those various points. And so I spoke before that we're prepared to scale up as necessary to meet the needs should we be fortunate enough to have a commercial product. But that absolutely will be the gating factor for actually giving drug. And that's been something we've been focused on for just about 5 years now.
Okay, great. Thanks, Joel.
Thank you.
I think that concludes our talk.